Good afternoon. My name is George Paris again. I'm in the middle of a series of videos related to my interest in evolution, development, evo, devo, etc. What I want to do today, first I need to put this in the context that everybody can understand. What has happened over the last 70 years probably is a study of biology which, when launched in the 1950s, people thought would lead directly to an understanding of evolution and development. But it really hasn't. I want to explain why. Okay, let's say you wanted to build a house. I show you a picture of a log cabin or a mansion or a skyscraper. And I say, here's your house. And you say, great. Okay. Then I take you out and I show you some tools. This would be hammers, nails, wrenches, jackhammers, cement mixers, stuff like that. And I show you a pile of building materials, which would be like concrete, steel, rebar, wire, cables, electrical lights, things like that. And I said, okay, that's it. here's your house. And you would say, well, these are the tools and materials used to build a house, but how do I get from the tools and materials to the house? And that's the same problem we have today. We have understanding of proteins and how proteins work, and we understand a lot of the biopolymers, DNA, RNA, protein, polypeptides, sugars, etc. But we don't know how to make the organism. It's not, if we can know everything we need to know about the tools, we can know everything we need to know about the materials, and still not know how to get from the tools and materials to the final product. Nor do we understand the evolution of how we would go from that log cabin to a mansion to a skyscraper. There's like an evolution of building here. So the same problem exists in biology that I just described in material terms. What I want to do today in a program and a lecture, I do lectures. In this lecture, I'd like to go, the title of the lecture, I guess, would be Pangenes and the Master Development Program. Okay. I specifically want to emphasize program, not plan. If I gave you a plan for a building, you still wouldn't have the building. In fact, if I gave you the tools and a plan and all the materials, you probably couldn't get to the building because you don't know the program needed to go from the materials to the final product. I mean, if I were going to build a building, I would end up by bringing in an excavator, dig a basement, drill a well, bring in some guys to put in install a concrete slab, some walls, and go up from that. It's a whole series of steps that have to happen in a specific order. And that's exactly what has to happen when you go from that single cell I was talking about in the previous videos to the final adult organism. And so knowing the proteins, knowing the materials doesn't get you there. And the question is, that program must be embedded in the genetic material. And we haven't found it yet. What I've done for the last 20 years, actually the first 10 of the last 20 years, was to look at how we could do that and develop a hypothesis about that could happen. I'm not going to reveal that hypothesis here. Actually have published this book, The Master Development Program Hypothesis, okay, for evolution and development. The funny thing about it was when I was working on development, I wasn't worried about evolution at the time. When I got finished with the process, came up with a hypothesis, I realized, hey, this would work for evolution as well as development, and I felt really good. But I don't know that that's, it's a hypothesis. It's just an idea from a crazy old chemist. In any event, let me go forward here and go through this. This is what I want to do today is primarily set the stage, show you where biology has gone from Darwin until today, at least to the early 2000s. There may be some stuff that's happened in the last couple of years I don't know about. But this is going to involve a lot of people and a lot of dates and whatnot. So I have my usual supply of notes I want to go through. And if we start with Darwin, you recall Darwin, was it 1860 something, published his book on evolution, natural selection. And the way his hypothesis for natural selection and evolution worked, you had a population that had variation in it. And then natural selection would determine which ones of those selections, those traits would be preferred. So he had to have both natural selection and variation in order to accomplish evolution. So he was, after talking about natural selection, he had to go discuss how to create variation. And at the time, there was not a lot of understanding of that. Basically, 1870-ish, Darwin introduced the idea of pangenes. I'll talk a little bit more about that in a second. And pangenes were supposed to be the source of morphology. Actually, I would call them traits that create morphology, physiology, biochemistry, those things. And between then and now, we've progressed substantially. But let's go back and start through this process step by step. The idea of inheritable acquired characteristics, which is abbreviated IAC in some books you'll read. Actually, it's sort of intuitive to people. Like if I'm a guy and I develop some skill, some strength, etc., I would like that to be inherited by my kids. Probably isn't, but I would like it to be. And so from at least the time of the ancient Greeks, the idea that you would have inheritable characteristics was accepted. It's sort of intuitive. People like it. Up until Jean-Baptiste Lamarck, and his birth and date were 1744 through 1829. And about 1800, Lamarck put this in terms that modern Western thinkers could recall. And his view was that the tendency is to go from less complex things to more complex things. This is a natural tendency. And the things that worked on that are use and disuse of organs and limbs and whatnot. And those obviously were tied directly to the environment, because the environment determines which organs and limbs you have to use in order to survive. And so this was his thought about how this works. And it was really the only thing, and it was really pretty well accepted by Darwin. Now, his acquired traits. Now, the one thing that I'll mention here is there was some general support, some observational support. I wouldn't call it statistical data or anything like that, but at least you would have some observational support. And in order for there not to be a lot of exceptions to that, he made one caveat, in that this trait had to be accepted by both the parents, both sex, the male and the female who created the offspring. If they were both excelling in one direction, then the offspring would excel. And what that did, just by chance, it avoided the problem that Mendel developed, where you have recessive and dominant traits, and so you didn't have to deal with that problem. So that's sort of how the system seemed to work. Let me see if I can change the page here. All right. Now, Darwin continued working, continued writing. And in his second volume of a book he called The Variation, or Variations, which was published in 1868, he outlined a process which was based on Lamarck's idea. Basically, he proposed that there's something called gemmules, and they're in all developing cells, according to Darwin. And they are influenced by use and disuse in the environment. And they create variation. There's some variation in the gemmules based on the environment and use and disuse. Those gemmules get circulated and end up in the sex organs of males and females. They are then inherited, and then natural selection takes over and creates species. That's the hypothesis that Darwin published in 1868. He had, I think it's his cousin actually, 1870, well, right immediately after that, a fellow by the name of Francis Galton, was, I think he was a cousin of Darwin, apparently decided he was going to support Darwin and prove his hypothesis. And he took rabbits of the same species, some white, some black, and he did massive blood transfusions, expecting that he would end up take the white line of rabbits and the black line of rabbits, and he would create, by swapping blood, where those gemmules are supposed to be moving, or at least he assumed, everybody assumed that's what it would be, would carry to their respective sex organs and the two different lines of rabbits the appropriate gemmules and cause this to happen. Well, it didn't. So basically, Galton proved, I think it was published in 1871, that Darwin was wrong. As far as I can tell, Darwin's concession here was he said, well, I never said it had to go in the blood. It might have gone in some other fluid. But that's sort of where it ended. Nobody got too excited about it. Now, this was sort of immediately followed up by August Weismann, 1834 to 1914. In 1884, he proposed something called the Germplasm Theory. And basically, he's reversing or undoing Darwin. He says, look, you have somatic cells and you have germ cells, and the two do not interact. They don't, there's no connection between the two. There's a barrier between the two. It's actually called the Weismann barrier. Okay, and so it basically, this put an end. Really, this 1884 put an end to the inheritance of acquired characteristics. After that point, nobody really believed that. Now, you recall Mendel, 1866, published a paper describing his experiments with peas, different traits, showing a pattern of genetic inheritance or pattern of inheritance. And he didn't just publish it. It was very obscurely published, but he took copies of the paper and sent them to about 40 different well-known scientists, mainly botanists and people doing work in this area. I believe Darwin got a copy, and also a fellow by the name of Negrelli got a copy. Now, Negrelli was a well-known scientist, 1817, 1891 time frame. And so he was the right age to get this document in 1866. And interestingly enough, apparently Negrelli actually interacted with Mendel and either discouraged him or put him on the wrong track accidentally. But it led Mendel into some experiments that didn't work very well, and he lost interest and went off and did other stuff. Now, Negrelli had a student named Carl Correns, who lived 1933, and that will be important later. But anyway, for the most part, there was no public, broad understanding or appreciation of Mendel's work. It sort of vanished. Okay, it was published and vanished because Mendel didn't pursue it any further, and nobody else was really pursuing it. Then we come to 1848 to 1835. That's Hugo de Vries. And he was working in biology, and suddenly he came across a plant called the evening primrose. And this evening primrose, he discovered, was creating a lot of variation. You know, one plant, lots of variations. He decided, I guess it was 1889, he published a document called the Intracellular Pangenesis, a important book. And he talked about the traits that were present in these plants, the morphology, the physiology, the biochemistry. And he said that these are due to pangenes, which is really, I don't know if Darwin, apparently Darwin's gemmules, he used apparently different terminology, including pangenes. Well, De Vries actually clearly identified pangenes and pangenesis, and they talked about intracellular pangenesis. Now, if you continue on until about 1890, sorry if I've got a little cold or something here. If you go to about 1890, De Vries started doing some experiments that were a lot like what Mendel had done. And he came to the same conclusion that Mendel came to. And along the way, he apparently found Mendel's documents. And how much Mendel influenced what he did, what he found, is an issue for speculation. I'm not going to go there. In any event, about 1900, De Vries published a theory. He didn't cite Mendel, but he basically had the same result. As a matter of fact, he appears to have used some of Mendel's terminology. Well, guess what? While people were starting to accredit him with Mendelian evolution or inheritance, this guy Correns, who was a student of the guy who had received one of the papers and probably got involved with that review and whatnot, comes up and says, hey, guess what? Mendel did this and you didn't cite him. This is a minor scandal. Apparently, De Vries very quickly acknowledged, hey, Mendel actually did this work in 1866, like 30 years earlier, and 34 years earlier. And so, you know, he didn't fight that issue, which is a good thing when you're caught red-handed. Instead, De Vries turned his interest to the other point that he had noticed, and this is that these plants mutated very quickly. These evening primrose, they didn't wait around for like 1,000 years to become something else. It happened like one generation. And so he invented or came up with the concept of mutation theory. He basically say, hey, things can change very quickly. They don't have to take thousands of years to happen. And it's called saltation, something turning very quickly in one step. So this was a new idea that was quite different than Darwin. I'll mention another fellow here, primarily for his overall influence, William Bateson, 1861-1926. Before 1900, he primarily had done work on looking at different organism species. And in Darwin's world of variation, you would probably expect the variation to be around, if you took some biometric measurement, like the length of something or the width of something or the weight of something, and you looked at a population, you'd expect it to probably fall into some sort of a normal distribution. Well, what Bateson realized is that some traits do that, and other traits are bimodal. There's like two different groups within the same group that have differences in a particular biometric measurement. That was an interesting observation. Around 1900, Bateson was supporting Mendelian evolution. He saw the value there. He saw the beauty of it, and he also accepted saltation, mutation. And he began to use the term genetics just to define what we were talking about. What's the field of study? Well, it's genetics. And so in the time frame 1905, 1906, the term genetics was introduced by Bateson. And so that he gets credit for that influence. Now, the term gene was not really coined until William Johansen, 1909. When you start working with this inheritance thing, you have genetics and pangenes, and so you need a vocabulary. And so one of the things which Johansen introduced was the idea of the gene, and also the idea of phenotype and genotype. Now, phenotype is, of course, what the physical body looks like, and the genotype is what the genetic underlying factors are. So this terminology was getting into the literature 1900 to 1909, something like that. Now, I want to make a note about technology here. It's actually very important. Up until this point, late 1800s, basically people were like looking at biological specimens and characterizing it at that level. But the microscope had been invented in 1674, and by 1830 you had compound microscopes, so people were starting to do quite a bit of microscopy. And around 1872, it was realized the relationship between light and the wavelength of light and the ability to resolve the size of objects. If you're smaller than the wavelength of light, you can't be resolved with a typical simple microscope. But something more important actually happened, I believe. Late 1800s, if you read some of my organic chemistry books and whatnot, you will know that late 1800s, there was a big influx of organic dyes, colored compounds that had economic value. And one of the ones that came out of that was something called methylene blue, which became used as a pharmaceutical for malaria. And one of the things that was obvious is that it stained cells, so that when you looked at them under microscopes, certain parts of the cells could be stained selectively and would show up better. So the combination of microscopes improving, being better understood, and staining, late 1800s, so methylene blue was invented, discovered 1876, so we're talking 1880-ish, when this all got rolling well. This is when people really started focusing on the cellular level of biology, okay, cytology. By the way, in 1903, ultra microscopes were invented, which could see below the wavelength of visible light. Visible light, by the way, the least we can see wavelength-wise is about 350 nanometers. That'd be 0.35 microns. A fellow by the name of E.B. Wilson started using the microscope extensively. He is credited with founding the area of cell biology. And he also got an interest in embryology. And one of the things he discovered at that point was he was looking at different organisms evolving or developing into their adults, and realized that some of them had similar patterns of development. And from that, he deduced that they must have a common ancestor. Okay, so that's not revolutionary interest, but it did have some interest and influence down the road. Now, Wilson had a student named Walter Sutton, and he had a friend named Theodore Bouvier, and he apparently interacted with both of them. This is in the time frame of 1900-ish. Lots of exciting stuff was happening in 1900. And so this guy Sutton, the student Sutton, his time frame is 1877, 1916. Looks like he died fairly young there. And then Bouvier was 1862 to 1915. In any event, out of their work and the new reliance on stained cell material in the microscopes, not only were we developing appreciation for the cell cycle, which is an important contribution, but also it was decided that the way that things were working in there, the chromosomes, colored, were the agents of heredity. Where the chromosomes went is where the heredity went. And so, in addition, if the chromosomes are the agents of heredity, then the changes in heredity must be mutations in the chromosomes. So that was established in 1903, 1902, that time frame. Then Wilson invited Thomas Hunt Morgan to Columbia, 1904. And in this time period, there were some people at Columbia doing fruit fly work, not on a large scale, but fruit flies. This was Drosophila melanogaster. Okay, they were convenient to work with because they were cheap to hold. Anybody could graze them. They were, easy to feed, and they did have some variation in them that people could find, changes in color, size, shape, etc. And so people had done some work, not big scale work, but they'd done some work. Around 1909, Morgan got excited. That's 5 years after Morgan arrived there, but 5 years later, he's suddenly sees the value of doing this. He had it may have been because he had several students who were very good, one of which was called Calvin Bridges. Bridges was like Mr. Fix-It, I guess. He was the one who developed the techniques for mass raising of fruit flies and for killing fruit flies and sorting fruit flies. And he had a very good eye for spotting, this 1 in 1,000 that had a mutation in it. And so Bridges was made a lot of contributions in that regard. Another student was Alfred Sturdivant. Sturdivant, 1891 to 1970. We're starting to get into our time frame. Out of this work, along the way, one of the things that Wilson had done, he'd realized how sex is determined in humans. X chromosomes, Y chromosomes. Males are XY, females are XX. Okay. And so it was apparent then, in looking at these fruit flies and the inherited traits, the mutations, sometimes they'd show up only in females, sometimes only in males, etc. And so it was apparent that some of these things were sex-linked, which would mean they were linked to certain chromosomes. And when that happened, Sturdivant got the idea and worked it out very quickly, apparently, like overnight or within a few days. Worked out a way to link chromosomes together. Basically, the idea was that he hypothesized that the genes are on, in a linear fashion, on the chromosome, and that all you had to do was to look at mutations and see which mutations transferred together. And if it didn't transfer together, there must have been a break between those. So using this as a method of exploration, he was able to physically link certain places to certain mutations on the chromosome. That was interesting. Now, Morgan, and of course there was papers published in scientific journals, and in 1915, armed with this information, the Sturdivant hypothesis and whatnot, Morgan wrote a book called The Mechanism of Mendelian Evolution, or inheritance. The Mechanism of Mendelian Inheritance. I keep screwing that up. But anyway, so that book really sort of put in total perspective the idea of genes, proteins, mutations, chromosomes, and Mendel's work with inheritance. Morgan had one more student of great note, a fellow by the name of Herman J Mueller. Now, Mueller was a theoretician. He wasn't a hands-on guy. In fact, apparently he was not very good in the laboratory. But he was very upset. He was frustrated because when the papers got written, the people with hands-on work got their names on the papers, but whatever contribution he made did not get recognized. He didn't get his name on the papers. I don't recall exactly what Mueller's history was beyond his graduate work, but Mueller lived 1890, 1967, and in 1926 he decided to try to create spontaneous or induced mutations. Now, up until this point, all the mutations that had been observed were just regular, something spontaneously happened. You looked at 1,000 flies and one of them had changed, and that was, you picked out your mutation. Mueller got the idea, maybe I can actually cause that to happen chemically or physically. And so he used various techniques, heating, cooling, all that sort of stuff, to see if he could cause mutations. That didn't work. He also tried chemicals, including arsenic compounds. That didn't work. But finally, now you got to appreciate what he's looking at here. He's not looking at point mutations. He's looking at mutations that cause major morphological changes or that you can see under the microscope. And most likely the mutations he's looking at are really gigantic chromosome scrambling type mutations where you have double strand breaks and recombination and all kinds of strange things happening. And so anyway, this is 1926, and he found that he didn't do it with temperature, he didn't do it with chemicals, but he did find that X-rays, which people were using pretty randomly at the time. Medical use of X-rays became very popular in World War I and beyond, and you could even get your feet fluoroscoped and stuff right up until the 1950s. But he pointed out the potential danger of that, and as a result, his story goes on. I'll probably pick it up later, but the idea that X-rays cause mutations and radiation cause mutations really became important because of the work during World War II on nuclear energy and the atomic bomb and whatnot. And so in 1946, he received the Nobel Prize for induced mutations, and his concern was primarily what this is going to do to the human population. What's it going to cause? How it's going to cause us to evolve? And he had a hypothesis that was based on his observations. His observations going back to cytology. He was looking at cells that had major changes in the chromosomal arrangements. He was hitting them with a very high energy X-ray or other high energy and causing big damage to the chromosomes. And if you look, if those are your criteria, then his hypothesis that there's a linear, no threshold dose of X-rays will cause mutations. In other words, if you hit it with enough energy to cause these massive mutations, there's no threshold of that. If you hit with enough energy with one dose, one event, then you can break up a bunch of chemical bonds. I've written a whole book debunking that when it comes to chemicals, but we'll deal with that some other time. Okay, so this takes us up until the period, if we go back and summarize everything that's going on here from, say, 1920 to 1960, that ends up being accumulated in something called the modern synthesis of evolution. Basically, we're trying to take, I keep confusing Einstein and Darwin, take Darwin's information and explain it in terms of some sort of mechanism. Along the way, evolution got changed from sort of what Darwin was thinking about to rather a change in the alleles of a protein. Proteins are obviously codes in the, it's recorded as a code in the DNA, and there may be variations in that code. And so that the variations, each variation is called an allele of that code. And so in that context, we're talking about evolution as changes in the protein codes. And we're focusing on proteins. That's very, very relevant, that we've reduced this down to the proteins. Now, natural selection is the force envisioned as that selects, it's the only force that takes the organism and adapts it to the environment. Okay. Mutations are in this modern synthesis are viewed as almost always adverse. We can knock out genes and they never work again. like the glow gene. We talked about that last time. Occasionally, though, it will create a new allele which has a little bit different function. And if you do this long enough, over a long enough period of time, as in Darwin's hypothesis, you get the slow progression from one species to another. You have morphological, physiological, and biochemical changes, and that ends up causing evolution, which leads to speciation. All right, so that is the modern synthesis, which was popular at least until the 1960s or something like that. Now, because of the focus on alleles, and obviously we're talking about protein coding genes, that has been the focus since that time period, the early 50s, 40s. Okay. Now, this brings us to two very important people, actually three or four. Francis Crick, 1916 to 2004, and James Watson, born in 1928. I think he's still alive, at least as of the time I'm doing this video in 2022. Wow. Okay. They were working together trying to figure out what the structure of DNA is. Now, deoxyribose nucleic acid had been realized for a long time, but DNA actually includes some proteins, histones, and stuff like that. And so it's like, how does this all fit together? They would not have been successful, at least it would have taken them much more time, had they not acquired some information from a lady by the name of Rosalind Franklin. Rosalind Franklin was the contemporary example of a female crystallographer. She wasn't the first female crystallographer, but several very important crystallographers were females in the 1900s, early 1900s. Rosalind Franklin was working. She lived 1920 to 1958. Short life. Doesn't get the credit that that longer life people get. And she was a specialist. She was a crystallographer, not a generalist, not a big picture person. But her data was critical. Her data, the interpretation of her crystallography was that there's a spiral. There's some sort of a helix going on here. With that information, Watson and Crick, who I believe there's a biologist and a physicist. So you got a biologist, a physicist, and a crystallographer, were able to come up with a structure of DNA. Now this is embarrassing. This should be absolutely embarrassing to every organic chemist out there. Okay. This is the classical example of the use of structural organic chemistry to solve a very critical, very important problem, the structure of DNA. Organic chemists had worked on structural chemistry since the 1870s, that time frame. But unfortunately, they had become fascinated with R.B. Woodward and some other people in the 40s and 50s and synthesis of exotic molecules. We're talking, things are personality driven here. And so while we were focusing on the work of R.B. Woodward and related folks, here these guys come up, take the principles that organic chemists had developed. They actually created models of a phosphate, the sugars, and the bases, and started rearranging them. And with the help of the crystal data from Rosalind Franklin, we're able to synthesize a model of what's called the double helix. Let me proudly show you my copy of the well-known book. This is I don't know if this is valuable or not. This is like an original edition. It's got, we have the photographs and whatnot in here. So anyway, that book, well, the paper was published in 1953. The paper, Watson Crick realized very quickly and made a note of it in their original paper that, hey, because we have this helix where we have interlocking chains, this gives us a perfect opportunity to explain how DNA gets replicated. Basically, the the chains come apart, and this chain is used as a template to build a new chain, and this chain is used as a template to build a new chain. All right. So the idea that these two chains would be templates for reconstructing the other half of the DNA was a very important point, and it was published along with the original structure. That was 1953. Of course, wham! Suddenly everybody's very excited about how DNA works. We have an important mechanism here, and so we're looking at how the codes in the DNA, of which they turn out to be 4 ATCG, how that relates to amino acids and how it relates to proteins. And so out of that work, starting in 1953, I have a very good friend, by the way, who I believe worked in that code breaking in the 1950s and 60s, a fellow by the name of Dolph Hatfield. Great guy, very guy. Look Dolph Hatfield up on Google. He has an amazing life. He retired from NIH a few years ago. Anyway, in 1965, Watson published something called it basically related DNA to RNA to protein, which became known as the central dogma of biology. DNA, RNA, protein. Again, we're focusing on a shiny object here. Okay, we have totally lost contact with the original work of Darwin and evolution. Crick came back in 1970 and said it's not quite that simple. Crick's still around. He was there long enough to realize that, hey, it's not, protein does not influence DNA and RNA, but RNA influences RNA and DNA influences DNA. So there's information transfer that is not straight linear, but proteins are the ultimate part there. Proteins are the tools of biology. That's not quite true, because RNA is also a tool of biology. If you go back, critical period between the chemicals and the first cells, RNA may have been the thing. In any event, we plugged along from 53 through 60, 70, 80, 1990s, and into the 2000s. Actually, about 1990, people said, well, gee, if we knew all those codes and all that chromosomes, we would understand evolution and everything else we need to understand. And so something called the Human Genome Project was started, multinational project. Laboratories, important laboratories in different parts of the world, Japan, Germany, Britain, America, France, other places, were able to sequence DNA slowly. A lot of hand manipulation and wet chemistry involved. But instrumentation was starting to catch up with this. And the project was moving very slowly into the late 1990s. It was projected that we would probably have the whole genome sequenced and understood in like 2050, 2040, 2030, something like that. Being a government program, it would have gone on forever. Okay. I'm down on bureaucrats. But anyway, in walks a fellow by the name of Michael Huckabeller. Who's that? That's not a name you probably recognize. He worked for a company that made sequencing machines and who was advancing the technology of gene sequencing, of DNA sequencing. And he realized by late 1990s that not only he had a sequencer that worked pretty quickly, but computational capabilities of computers had advanced tremendously. And so what most people were doing, these different research groups around the world, they were taking some marker, something important, and they were trying to work around that, gradually expand their turf, and eventually they were thinking in terms of linking this all together. But that would just take forever. What Huckabeller figured out was that if you if you look, if we got like something like 3 billion base pairs in our DNA, statistically, if you have 16, I think it's in sequence, around 16 individual base pairs, if you know what they are, that's probably statistically, it's basically unique. That sequence 16 long is pretty unique. It's not, the odds of finding it randomly is very low, unless it's intentionally repeated. So he said to himself, I guess, if we took the genome, the whole genome, and just broke it up, which we can do, we could chop this thing up into pieces. And then if we sequenced every piece and dumped all that data into a computer, we would just let the computer match up the 16 base pair units. And we could stitch it together virtually as opposed to physically. And there's some business acumen going on here. Basically, to do this, knowing that the government's out there doing it, and knowing that these guys sell the hardware to do that, they said, well, gee, if we were doing it quickly, we could probably sell a lot of our equipment to the government and to all these research groups that are supported by government contracts. And anybody else, like pharmaceutical companies or anybody who wanted to do this type of work, would benefit from our machinery. They created a company called Celera. It was spun off as a, quote, tracking stock. The company, Applied Biosystems, it was actually a spin-off or subsidiary of PerkinElmer. Applied Biosciences essentially had a tracking stock called Celera, Celera meaning speed, and they needed a flamboyant guy to run it, and they went and got J. Craig Venter to do that. And basically, I think they may have thought, well, eventually we'll sell this data to industry, but somewhere along the line they completely said, hey, we don't need to do that. We're just going to sell the hardware. We'll sell the technology to all these people who want to do this, and we'll give away the data, which created a very strange idea. This was the idea that the company Celera, which was on the stock exchange, you could buy the stock, was going to generate this data and give it away. Think about that for a while. In any event, the company essentially challenged the feds, all the academics out there who were doing this, primarily supported by federal or money or state money, whoever's governmental support for this, to essentially, we're going to do it first. So this got a very exciting period of time started. And if you ever want to follow this was very exciting. This is another book you should have. The Genome War. Okay, this goes into a lot of the nitty-gritty between Craig Venter and the NIH and all those guys. And that's a great book, by the way, very exciting to read. And so anyway, over a period of time, the stock got very juiced up. I mean, it started out at a couple of dollars a share, and I forget where it went to eventually. I did not see the business model of Celera. It just made no sense to me. However, there were other companies not related business-wise, but in the same area, that were patenting genes. I thought that might be a good idea, so I invested in one of those. I bought a bunch of stock at $16 a share and sold it at $240 when the genome was revealed in 2003. Celera went from, if I'd got on the Celera bandwagon, I could have doubled my money or tripled my money, at least. I remember I was selling at $4 a share. I said, I'm not going to buy that stuff. They have no business model. My mentality was you've got to actually have a product. Just relying on people buying it because it was speculative was not in my thought. But people do that, so you live and learn. Anyway, so Celera, I don't know if it exists anymore. I know it went through many changes. It's not a big deal. Interestingly, I'm here in Gaithersburg, Maryland, and they were in Rockville. And actually, I worked up and down the Rockville Pike. And at one point, there's actually a traffic camera that was on their building or right at their building. You could see people coming out of their building with the traffic camera, and I was like watching Celera through on the traffic camera. But interesting history. They got rich quick and then dissolved basically because they had no product. They gave away the one thing that they generated. Now, if it had been a business model that made any sense, first off, I wouldn't have known about it, because if the business model that would have made sense for Celera would be to not tell anybody instead of creating this war that they did with the federal agencies and whatnot over who's going to get the genome first. Rather than doing that, had they kept it all secret, done it very quietly, generate the genome, and then get a meeting with major pharmaceutical companies and walk in and say, first get everybody to sign confidentiality agreements, and then say, look, see this big book? This is the human genome. We've sequenced it. What are you going to pay us to have access to this? That would have made them some money for the data, but they never did that. For whatever reason, they just dispersed it. So anyway, up to this point, it was assumed that once you know the proteins, you would be able to understand pretty much everything about biology. You could understand all the proteins, all the once you know the genes, you could get all the proteins, you could get all the functionality. You would know how the whole genome works. But, you know, it was assumed people were talking about 1 million genes, 100,000 genes. It turned out that the whole human genome only has about 20,412 genes. These are protein coding sequences. People started talking about junk DNA. All this other DNA, which is most of the DNA, must be junk. It doesn't code for anything. And then people started thinking, well, maybe we don't have the whole picture here. Because what had happened, going back to my original analog, the proteins are tools, and sometimes they are materials as well. But basically, the functioning proteins, the enzymes, are tools, and some of the proteins, some of the polyamides are functional, or materials, building materials. They're not functional except as building materials. And so you have your tools and you have your building materials, and you can see the product. You can see me, you can see fish, you can see birds. You now have the genes, you have the building materials, you have the tools. You're still not there. You don't have a clue how these things cause that to happen, and you don't have a clue how the building went from being a log cabin to a mansion to a skyscraper, which is evolution. And so this is all kind of exciting. And that's really where I came in. I was, like I said, if you go back to some of my early videos on the subject, at that particular time in my career, I was like sort of out of work and unattached. And I saw this as a whole different problem. And what is missing here, and I think it's still missing, I don't think anybody I haven't seen a hypothesis except mine. But what is missing is the master developmental plan, actually program. I keep correcting myself, program. It has to be a program. It's not a plan. I can give you the floor plan of a building and say, tell me how to build it, or tell me to build it. And the plan itself doesn't tell you how to build it. You have to have a program, a sequence of events that go from the tools and materials to the product. So it's a program, not just a plan. And so this master development program is what I became interested in. Now, since we have sequenced, quote, the whole genome, we actually haven't sequenced the whole genome. They realized, especially this technique that was being used by Huckabeller and Venter, that if you get into the heterochromatin, that is the paracentric heterochromatin, which is always there. It's not, it's constitutive as opposed to facultative or whatever. It's very repetitive. And suddenly you couldn't use that same technique to assemble that material. As a result, they really didn't sequence that. They just, the euchromatin has been sequenced pretty well, but the heterochromatin hasn't. And it is my hypothesis that there is where the master developmental program exists, and how to get it out of there and how to make it work is a question that has not been addressed, I don't think, by any major people. Of course, I got interested in Evo Devo, started looking at Evo Devo and wondering, like, how does this work? Don't people understand this? And the only hypothesis that had come out that had any following, I believe, at all, had to do with essentially establishing gradients. There's some examples of people, a gradient of chemicals that would cause sequential development of segments and things like that. As a chemist, I look at that hypothesis, and we're talking about forming gradients in biological tissues. It just doesn't make sense. If you start looking at the precision of the organism, we actually have, you can go buy books of anatomy, and you will find that in these anatomical drawings of, say, human beings, you will find exact blood vessels, exact branching of blood vessels, right down to fine detail in these people, and they're the same in all people. The anatomical drawings of that you find in these catalogs exist because humans are all pretty much the same. The fine detail, you cannot create that and maintain it. It doesn't matter what the food they were eating was. It doesn't matter where they lived. It doesn't matter how cold it was, how hot it was. The history of a mother who's running around with this fetus inside of her doesn't exist. Chemical gradients are going to be changed by compression, temperature, all kinds of things that would affect this, and that just is not a suitable examination or explanation for why this morphology, physiology, and biochemistry exist. It has to be much more fundamental. It has to be pretty much hardwired. If you look at identical twins, it repeated twice. Exactly. You can't get that with chemical gradients. The whole developmental process, from even after birth, there's still development going on. And even a couple of identical twins are not going to be exactly the same environment all their lives. And so if there's chemical gradients, the precision of development cannot be explained with chemical gradients. It has to be otherwise explained. It has to be hardwired in the genome, and it appears to be tucked into the heterochromatin. Now, why would you do that, by the way? Well, if it's in a heterochromatin, it's very hard to disrupt it. And if you have a species, you don't want your morphology, physiology, and biology disrupted. The proteins are just there as tools. Don't really play a direct role in development. They are the tools that are used to develop, but they don't control development. So anyway, if you want to know that, where I believe that went, you have to have another book. This book. This is available on Amazon.com. I recommend it. My next video, I've got some more work to do in leading up to this, but eventually I'll get to discussing this book in some detail. But anyway, thank you for your time. I've been here just about another hour. Worked out well. And you'll notice I basically do lectures. This is not a scripted presentation. It's sort of like how I feel today is how it went. And I'm doing it in my basement. So I get interrupted by my cat occasionally, and my family comes in from time to time, and all that sort of thing. Anyway, thank you for your time. If you like the video, please indicate so. Tell all your friends about it, and feel free to forward it to anybody. This is free of charge. If you want to give me money, buy the book, all right, which is very cheap. It's like probably $15 or less. I don't know. Have a great day. Thank you for your time.